Today, I'm going to talk about corollary discharge signaling and repetitive behavior in children with autism spectrum disorder. Previous study has found that sensory hyporesponsiveness is related with repetitive motor behaviors. However, it is unclear what exactly causes these symptoms. This study aims to examine corollary discharge signaling as a potential underlying mechanism. When we are about to make a movement, the motor area of our brain sends out a motor command to the corresponding muscles to carry out the specific action, and that will result in certain sensory input. At the same time, it also sends out a corollary discharge signal that is like a copy of the motor command, but is sent to the sensory neurons instead, so they can make a prediction about the incoming sensory input. By utilizing such predictions, we can suppress the sensory consequences of self-generated actions. For example, that is exactly why we cannot tickle ourselves. From now on, I will refer to corollary discharge as CD for short. Now imagine if something goes awry with CD signaling, then the sensory neurons will not be able to make a correct prediction of the incoming sensory input, which can lead to abnormally salient sensory consequences of self-generated actions. This may lead to a reduced attention to external sensory input. Behaviorally, that may look like sensory hyporesponsiveness. At the same time, when you are under-aroused by external sensory input, you may look inward to generate enough sensory input through your own actions, resulting in repetitive motor mannerisms. Therefore, we hypothesize that abnormal CD signaling may underlie these two core symptoms of ASD. For the sake of time, I will only talk about repetitive behaviors in this talk, but we have data on sensory hyporesponsiveness too, so just send me an email if you're interested. To measure CD, we used the blanking task in this study. The task goes like this. Each trial starts with a red dot in the center of the screen. Participants are supposed to fixate on it. Soon after, the dot will change color to black to signal it will move to a different location to the left or to the right. At the pre-saccadic location, participants are told to follow the target as soon as possible. And once a saccade is detected, the target will shortly disappear and jump to a different location, either to the left or to the right. We call that the post-saccadic location. These dotted circles are just placeholders for us to understand the task. Participants do not see them on the screen. Now at this point, participants to answer a simple question, the direction of the target jump. In this case, the target jumped left, so left is the correct answer. However, our saccades are often not so precise and can often fall short of the target. For example, on the same trial, let's say participants' eye landed here, and we call that the saccade landing site. If they have intact CD, they will know that their eye fell short of the pre-saccadic location. And by using that information, they can give the correct answer that the target jumped left. But if they do not have access to the CD information, then they cannot know that their saccade fell short and may rely on the saccade landing site as a proxy of the pre-saccadic location. In this case, the target is to the right of their eye, so the answer jumped right. However, that answer is incorrect. So this is how we measure oculomotor CD. And previous study in healthy adults found no relationship between landing site and perceptual judgment, suggesting that participants had access to intact CD information. In this study, we used a different analysis approach to look into nuanced interactions between landing site and CD. 31 ASD children and 35 typically developing children completed the blanking task. TD group had higher IQ than ASD group, but the two groups did not differ significantly on age, sex, education, or saccade metrics. We used multi-level modeling to predict participants' response on each trial. Here, forward means the target jumped further away from the fixation point, and backward jumping towards the fixation. I will now go through the predictors one by one. Target displacement means how much the target jumps forward or backward. It may be an obvious jump, or it may not move at all. In theory, target displacement should be the strongest and only predictor of participants' response if they have intact CD, regardless of errors in Scout landing site. The bigger the forward jump, the more likely they will answer forward. But if they don't have reliable CD information, they will be less sensitive to the target jump, shown as a flatter slope in this graph. Meanwhile, they may rely on other information such as Scout landing site, and we broke that down into error direction and error size. Error direction refers to whether the Scout landing site falls short of or goes over the pre-saccadic location. When the eye falls short of the target, it's called a hypometric saccade. When the eye goes to the pre-saccadic location, it's called a hypermetric saccade. If participants have intact CD, there should be no relationship between the error direction and response. Whether their saccade is too short or too long should have no effect on their judgment of the target jump. However, if participants have impaired CD and therefore rely on error direction to respond, we may expect a low likelihood to answer a forward jump when their saccade is too long. Because as the saccade landing site falls ahead of the pre-saccadic location, it is more likely for the post-saccadic location to be backward to the saccade landing site. If their saccade is too short, then it is more likely for the post-saccadic location to be forward to their eye, and we would expect to see a high likelihood to answer forward as a result. Lastly, error size refers to the distance between saccade landing site and pre-saccadic location. The smaller the error size, the more accurate the saccade is. Now let's look at the actual results. As expected, we found a very strong effect of target jump across all participants, suggesting that overall, participants were utilizing CD information on this task. In this graph, the blue line is the ASD group, and the pink line is the TD group. The slope of the ASD group is slightly flatter, but the difference between the two groups was not significant. Next, we found an effect of error direction. In this graph, the blue bars are the ASD group, and the pink bars are the TD group. Indeed, we found that participants were more likely to answer forward when the saccade is too compared to when the saccade is too long. However, the odds of forward response was not significantly different from 1 in either condition. In other words, across all participants, although there was a slight indication of using saccade landing site instead of CD to make perceptual judgments, there wasn't a strong bias of answering forward or backward on average. Lastly, we also found a target displacement by error size interaction that has not been explored in previous studies with healthy adults. In this graph, the green line is when the error size is large, and the red line is when the error size is small. We found that across participants, when they made more accurate saccades, they were more sensitive to the target jump. But when their eyes fell further away from the pre-saccadic location, they became worse at judging the target jump. One interpretation is that when the eye is further away, the spatial uncertainty of the target becomes higher, and participants may use non-CD information to help them respond. Although we did not find any group differences in participants' reliance on CD information, we continued to explore our hypothesis on the potential relationship between CD signaling and repetitive motor behaviors in ASD. We included the restricted and repetitive behaviors domain score from the ADI into the model. ADI is a standardized semi-structured clinical interview conducted with caregivers that measures current and past restricted and repetitive behaviors. We took out saccade direction from the model because it was not a significant predictor of task performance in Model 1. We found a repetitive behavior by error direction interaction. A quick reminder, if participants have intact CD, there should be no relationship between error direction and response. But if they have impaired CD and rely on error direction instead, then we expect to see a high likelihood of forward response when the saccade is too short, and a low likelihood of forward response when the saccade is too long. Breaking the interaction down by high and low values of ADI revealed that the effect of error direction was only significant in ASD participants with severe repetitive behaviors. In this graph, the dark blue bars are the group with severe repetitive behavior, and the light blue bars are the group with less repetitive behavior. Specifically, ASD participants with severe repetitive behaviors were more likely to make forward judgments when their saccades were too short, but not on saccades that were too long. In other words, when their eyes fell short of the pre-saccadic location, the post-saccadic location was more likely to fall forward of the saccade landing site, and they were more likely to answer forward in this case, suggesting that they were relying on saccade landing site instead of CD to make the perceptual judgment. In conclusion, we demonstrated that children were able to perform the blanking task in a similar fashion as adults, so this could be a useful experimental paradigm to measure CD signaling in children. We found that there were no general group differences in task performance. However, ASD participants with more severe repetitive behaviors did exhibit a tendency to rely on non-CD information, suggesting that abnormal CD signaling may be a potential mechanism of this specific symptom. Therefore, potential future interventions aiming to enhance the link between action and perception may be a targeted treatment for repetitive motor mannerisms in children with ASD. Here are the references. And lastly, I'd like to thank the founding agencies, my advisor, the research team, and the participants. Please feel free to send me an email if you have any questions. Thank you.